When we prescribe medications to patients, we keep telling them, you need to take the pills regularly. But we know that committing to a lifelong medication on a daily basis for blood pressure or blood sugar or cholesterol is not always easy, and a sizable proportion of our patients will abandon the medications after some time. But is it possible that medications continue to work even after the patient has stopped taking them? Do they retain their effects even when they are not detectable in the blood? I asked this question to the AI, and that was the answer.
It's unlikely that most regularly taken daily medications would continue to have a therapeutic effect 6 months after cessation. However, some medications, especially long-acting injectables or medications that cause lasting changes in the body, can have effects that last for months.
But how accurate was the answer of the AI? Let's see together.
Welcome to Cardio Buzz, your one-stop shop for all things cardiology. We bring you the news, groundbreaking research, and the most recent guidelines. We interview the experts in the field and try to answer the most difficult questions of everyday practice.
The impact of a person's actions, even after they are gone, is similar to the legacy or an inheritance that's passed down through generations. Similar to humans, do drugs have a legacy? Do they have long-term benefits even after discontinuation? Let's see what happens with diabetes, cholesterol, and blood pressure medications. Let's start by diabetes. In a famous study, the UK PDS study, that randomized patients with type 2 diabetes to either conventional sugar control versus intensive sugar control. In the active phase of the trial, which lasted 10 years, the intensive sugar control group had a lower risk of diabetes complications. But after the trial ended, patients in both arms were recommended to start the intensive blood sugar control. Patients who were in the conventional arm were shifted to the intensive arm, and those who were on the intensive arm maintained the intensive therapy. As a result, the blood sugar concentration in both groups converged after 1 year. And despite having almost identical blood sugar levels, the reduction in the risk of microvascular disease that was seen in the early study in the intensive control group persisted over 10 years post-trial follow-up. And the reduction of microvascular disease was also accompanied by a reduction of myocardial infarction and death. This long-term protective effect of early intensive therapy was given the name legacy effect. The earlier, tighter control of glucose levels in the active group during the trial also generated health benefits years and years later. In another study on diabetes, the STENO-2 study, the trial looked at patients with type 2 diabetes and microalbuminuria. For 8 years, they were assigned to either conventional care or intensified care, which included, in addition to treatment, behavioral modifications, lifestyle interventions, and pharmacological approaches. After 8 years, the study transitioned just to observation, with all patients, including those in the control group, shifted to the intensified therapy. The STENO-2 trial demonstrated that intensive therapy reduced the progression to diabetic nephropathy by 48%, and this was seen during the follow-up period that extended to 21 years. In hypertension, we know from the trials of antihypertensive medications that patients who used these medications got a reduction in overall mortality even after the trial ended, when most patients in both arms were given the active therapy. These findings were robust and consistent with different indications and different medications, including beta blockers, ACE inhibitors, and diuretics. Interestingly, the TROPHY study showed that candesartan, which is another antihypertensive, effectively reduced the development of hypertension in individuals with pre-hypertension when it was given for 2 years. Moreover, this protective effect persisted for an additional 2 years after the medication was discontinued, suggesting again the legacy effect. So candesartan proved to be useful 2 years after it was discontinued. In hypercholesterolemia, randomized clinical trials of lipid-lowering agents compared to placebo or usual care with follow-up after the randomized phase also demonstrated persistence of the benefit after the trial was over. Perhaps the best example comes from the 4-year trial on the injectable Repatha or evolocumab. The 4-year study was one of the biggest studies in cardiology. Patients were randomized to Repatha versus the standard of care. After the trial ended, there was an open-label extension, where the trial was extended and patients who were on placebo were shifted to Repatha. On follow-up, the arm that was initially on Repatha continued to show benefits compared to the arm that was initially on placebo, then shifted later on to Repatha. And the benefits were 23% less risk of cardiovascular death. Those benefits were seen 5 years after follow-up. In kidney disease, deterioration of renal functions is scary. Now we have at least 4 drugs to slow down the decline in kidney functions. Standing out among them is empagliflozin. In the EMPA kidney trial, the benefits of empagliflozin in patients with chronic kidney disease were amazing. Empagliflozin slowed down the progression of chronic kidney disease, but what happened after the patients stopped taking the drug? Did these benefits just disappear? Well, no. That's what exactly researchers wanted to find out in the post-trial follow-up, and the results are pretty remarkable. They followed patients from the original IMPACT kidney trial for 2 more years after they stopped taking empagliflozin, and the results were published in the New England Journal of Medicine. During the post-trial follow-up, patients were not systematically given empagliflozin, but local practitioners could prescribe open-label SGLT2 inhibitors, and that was in 40% of patients in both groups. Even after the trial ended, there was still a trend towards benefit, especially in the first year, and the benefit was quite significant. Even after empagliflozin was discontinued, patients who had initially taken it still had a significantly lower risk of kidney disease progression or cardiovascular death. We're talking like 21% reduction in risk of kidney disease progression to end-stage renal disease and cardiovascular death. The benefits were consistent across all sorts of patients, regardless of whether they had diabetes, their eGFR levels, or the cause of the kidney disease. Basically, empagliflozin helped everyone. So you might be wondering, how can a drug continue to provide benefits even after we stop taking them? This is the legacy effect of drugs. It's like a snowball. The initial intervention sets in motion a cascade of beneficial changes that continue to unfold. So what's the explanation of this legacy effect? How does the drug continue to work although it's not detectable in the blood? There are 3 possible explanations. Number 1, disease regression. The long-term pathological changes in the affected tissues and organs can be reversed. Persistent elevation of blood sugar is known to increase glycosylation of proteins and induce oxidative stress. Hyperlipidemia will induce inflammation and plaque formation. Hypertension will induce vascular hypertrophy, rigidity, and fibrosis. These alterations can be avoided with early and thorough treatment and can be even reversed, which means they bring the clock backwards, and this results in better long-term outcomes. The second explanation is the effect on genes. Environmental factors can have effects on our genes. These are called epigenetic modifications. Epigenetics is like a set of instructions that tells the body how to use the information stored in the genes. Think of genes as a big book with lots of recipes. Epigenetics is like the sticky notes that you put on the pages to say which recipes to use and when. These sticky notes, epigenetics, can be influenced by lifestyle or by drugs, and these epigenetic changes can persist. Exposure to hyperglycemia, hypertension, or hypercholesterolemia at the cellular level can cause changes in DNA methylation and gene expression and in the signaling pathways, and these changes may persist even after these risk factors are normalized. Therefore, complications in older age can be seen as a legacy of exposure to the risk factors at young age, and conversely, drugs can also favorably modify the epigenetics. Statins, for example, preserve the length of the telomeres. In our DNA, telomeres are protective structures at the end of the chromosomes. They shorten with each cell division, acting as a kind of a molecular clock. Telomere shortening is closely associated with aging process and is considered a key marker of biological age. Statins, by preserving the length of the telomeres, may have potential effects on vascular aging and longevity. The third explanation is by the prevention of non-fatal events. Statistically, a patient who gets an MI or a stroke secondary to hypertension or hypercholesterolemia is more likely to get a second event compared to those who never got an event. So preventing the first event gives it a bonus of preventing more events compared to non-treatment or delayed treatment. In kidney disease, empagliflozin may preserve the number of the nephrons during the active trial period, therefore reducing hyperfiltration and kidney damage and the risk of end-stage renal disease on longer follow-up. So finally, the answer is yes, medications have a legacy effect. We see that with early intensive treatment of chronic diseases. These medications have beneficial effects not only on the pathology, but also on the genetics, the cellular memory, the vascular function, and structure. But a word of caution here, not all drugs have this legacy. For instance, antiplatelets, anticoagulants do not have the legacy effect. So what are the implications? Of course, this is not a reason or a call for patients to stop taking their medications and just rely on their legacy. Instead, this is a call to start treatment early. The earlier we start, the bigger the benefits. Each day passing with an elevated blood pressure, uncontrolled blood sugar, or an unhealthy lifestyle can have a long-lasting effect on our health. Investing early in health is like investing in wealth. Even with seemingly small amounts can lead to significant wealth and health accumulation over time. It's like the compound interest in retirement savings. It's also a call to stay on the treatment as long as you can. The legacy needs to be built over years or decades, not over weeks or months. The longer the treatment, the higher the expected benefit. Again, like the retirement fund. We got the legacy effect of candesartan after 2 years of continuous use. We got the legacy of Repatha after 2 to 3 years of use. And we saw the legacy of empagliflozin also after 2 years of use. 8 years of commitment to healthy lifestyle will give benefits for 21 more years.
Thank you for watching this episode. Don't forget to hit the like button, subscribe, and share the episode with your colleagues. See you in the next episode.